Okay, so this is the first lecture of the second part of the course, and we're going to start to look at bioinformatics here for the next few weeks. Okay, so um, I think we're all pretty familiar now with this little cartoon of the data science cycle. Uh, so we're going to focus a little bit upstream now of that cycle, which is these bioinformatics databases and portals and repositories. And we're going to take a look today at a few of them in some detail. So bioinformatics, let's go through our definitions just again to make sure we're all on the same page. I would say bioinformatics is the development of tools, databases to make biological information available to all life science researchers, okay? So that's definitely different than data science. And I think we have an intuition of what data science is now, and that's more about playing with the data set and trying to, dig into it with visualization and models. Here, of course, we need data, and I think you're all going to find that you need to reach out to the community and grab publicly available data and bring it into your session, into R, for example, and combine it with your data that you may have generated locally, right? And you go from there. So this is actually a very difficult step to go from a database, external database, into R. And in the goal, our goal here, the grail, is to get the data from outside into a tibble, into this table-like data structure that we've looked at quite a bit now. And that process of going from some external data source into your tibble, that's called wrangling. Some people might call it munging, and I think munging is a better word because honestly, this is like a really terrible thing to do. It's horrible, right? It's trying to learn somebody else's format and work that into what you think is reasonable ends up being a lot of work. Now, computational biology, you might say, I might ask, where does that fit in? And computational biology again is sort of, in my eyes, building software, say, or statistical technique for finding genes in a genome or predicting the three-dimensional structure from a nascent amino acid polypeptide, right? It might be something like trying to predict binding sites. So it's a specific problem that a computational biologist has built a technique for. And so as a data scientist, you're using those tools in your modeling procedure. You're asking like, well, where are the genes in my genome? What does this protein look like in three-dimensional space? So here, bioinformatics, data science, and computational biology are really three interrelated but distinct concepts. Okay, so bioinformatics can get very complicated very quickly. Okay, and it may not be completely clear why it's the source of complexity. So I thought I would step back a little bit and try and give you an intuition about what the central issues are, okay, and why things become so difficult at times. I find it quite difficult to use some of these databases out there. Just going to move this out of the way. Sorry about that. Okay, so we have to organize our thoughts. So I organize it along the central dogma or the DNA-protein axis. Okay, DNA, RNA, protein. I guess this is as good as any place to start. So let's consider just DNA for a second. All right. Now I want you to do a thought experiment here. Imagine that you were a bioinformatician, which you will be, and your goal was to make all biological data available to interested researchers. So the question here is, well, what does that look like? What are the issues that you have to consider? Okay, so for DNA, you definitely would have to record genomes. For what? Well, I guess for all sorts of different kinds of organisms across the tree of life, phages, viruses, archaea, bacteria, eukaryota, so the very small genomes, a few thousand base pairs, up to very large, tens of billions. Even within eukaryota, for example, you have this issue of then different species, taxa within eukaryota. even ancestral extant, these are extant organisms, but you'd have also extinct dinosaurs, for example, genomes to be considered. And then when you go into human, well, you have which human, right? One individual of 7 billion individuals. As sequencing becomes more and more common, those databases have to make those genomes accessible with proper, and it gets into issues of security and privacy, those kinds of things that complicates bioinformatics for sure. Government laws for protecting and for privacy, etc. But then even within one individual, is it just one genome of 3.7 billion base pairs? I don't really think so. I mean, we all have different clonotypes. Certain regions of our genomes are variable. In fact, even our tissue, our genomes differ a little bit from tissue to tissue. Some polymorphisms, nucleotide polymorphisms, or what are called CNVs, copy number variants. Okay, they vary from individual from tissue to tissue. Tumor, if you're unlucky enough to get cancer in your lifetime, these days we sequence tumors all the time. It becomes part of clinical practice because it tells us something about the neoplasticity. Genomic instability is a hallmark of cancer. Okay, so it means that the genomes are evolving very, very quickly. It's very dynamic. Okay, and the genome, those cells are mutating, amplifying regions, and loss of heterozygosity. So those events tell us something about how to treat the patient, or that's the dream. So, but then even in a tumor, are we talking about the primary tumor, or even which location of the primary tumor? That matters, because they're, geospatially, tumors are different depending on what part of the tumor you're looking at, the necrotic region versus the margin, the microenvironment. So, are we sequencing the cells around the tumor, the immune cells, the fibroblasts, the adipocytes? Because we found that those also inform on the tumor, the cancer itself, the nature of the cancer, or the metastases. So it may have been a breast tumor, but it's metastasized to kidney, liver, lung, brain, etc. All of these different sites would be sequenced. Okay, so I hope to convince you there that even within one tissue and one tumor, there's multiple genomes within one individual, of many individuals within one organism, of hundreds of thousands, if not millions of organisms across the tree of life. Those databases have to consider all of that. Now, in addition to that diversity, you have all sorts of issues about things like errors, right? So sequencers make errors, right? So those genomes get updated all the time. And if you did the required reading, yes, I hope you have, and you read the section on RefSec, you'll see a nice history about sequencing the yeast genome and how different labs contribute to that sequencing process. And so how do you put those together? Who's responsible for combining all the different parts of the yeast genome into one sort of consensus yeast genome? If there even is a consensus genome across the community. You have different assemblies, so depending on the sequencing platform and the depth, etc., you get different answers. We're not really sure which ones. You think of something like COVID-19 virus, it evolves quickly. All viruses evolve, I guess. Viruses evolve quickly. So, we need to keep track of that. And not only do we just want to keep track of those different strains of something like COVID-19, but we want the phylogeny, right? We want to know how those viruses evolved in the population. So how when we look at the polymorphisms in a virus, it tells us how it spread in the population. And that can be really useful information, as I'm sure some of you seen even in the public, in the kind of mainstream literature these days. What am I saying? New York Times, mainstream media, etc. They talk a little bit about the evolution of COVID-19, and even things like cell lines in the lab. They drift, right? They're not completely stable genomes. So there's a lot of things to consider just for DNA. And if we come back up here and we zoom out, you know, what do we mean really besides the genome? We want to know where the genes and the exons are, the promoters, the binding sites, enhancers. You know, so these can be 100,000 base pairs upstream or downstream of a gene, and they affect chromatin or the DNA superstructure. These are all different things that we need to keep track of for each one of these genomes, essentially, right? If we knew that information, we would want that. Now, the important thing here, and this is where I actually hit a little bit of detail, is that if we go back to this slide, I said bioinformatics goes straight into here, and you know, bioinformatics is raw data, etc. Well, you know, there is a certain amount of computational biology that is run on these data sets already that we wrangle into our data structures. Okay, so as a concrete example, gene finding. So, you know, gene finding in humans is not a pure computational process, of course. It's been a process of investigation by a lot of people over many, many years. Now, computation plays a role in finding those genes, but it's touched up and manually curated. It would never be advisable to download the human genome and try to predict all the genes from fresh. You wouldn't do that. You would download the predicted curated genes from the relevant data website or bioinformatic repository. But the point here is that genes and promoters, binding sites, enhancers, structure. Now the modeling is in the data. It's actually not necessarily raw data that comes straight from a sequencer or a mass spectrometer or whatever technology. It's actually been processed by people and made available. And those things are modeled, so they'll change, right? So here's a little caveat, right? I mean, almost or less than 20 years ago, we believed that there might have been over 100,000 human genes in our genome. Sorry, genes in the human genome. And now I think the community is still not in complete agreement, but there's probably between 14 and 20,000 genes. So I'm not sure how, you know, we went from 100,000 down to 20,000 in the space of 20 years, but I think that does show you that, you know, our what we believe, the structure of the genome is changing, right? And those databases, those bioinformatic repositories, have to, you know, adjust to that change, right, and track it. You know, RNA, well, you know, one gene, but many isoforms because of alternative splicing. Not all alternative splicings are you know, observed in nature. We have to keep track of those transcripts and isoforms, right? Small RNAs like long non-coding RNAs or microRNAs, they play regulatory roles, so they're actually small RNAs that fold. They form three-dimensional structure, so we would want to track that. That's partly modeling. Sometimes it's structural, like NMR or crystallography, but that information has to be put out there. And it gets into something else here of describing regulatory relationships. So a small microRNA might, so what they do is they bind to mRNAs and they, you know, can basically blow it up, right? Cut it. So that's a relationship, right? That this microRNA interacts with a specific mRNA and controls its expression. So how do you start to describe those regulatory relationships between the objects, right? Between microRNAs and transcripts, or between genes and transcripts in the form of alternative splicing, etc. And there's a lot of uncertainty there. These are models. And cooperative behavior I'm referring to here is that not all the time are these relationships so binary. It's not always just one microRNA and one gene. It's sometimes an ensemble of small RNAs and a transcript, or proteins that form a complex, or with cofactors, etc., to bind DNA, etc. So how do you start to describe those higher order interactions, right, that exist out there? Okay, so I wanted to say here is that, you know, you have one individual with thousands of transcriptomes. Now this is a really important concept because, even if we have maybe one genome, you'd say, well, Mike, all those things that you described down here, well, okay, but it's all basically the same genome, right? I give you that there's variability, but now that's certainly not true at the transcriptional level when we talk about transcriptome. So transcriptome would be the complete collection of transcripts. So you think about taking a snapshot of a cell at a moment, and you count all the transcripts across all species, across all genes, and you say, okay, well, that's this transcript and this transcript are upregulated, and these guys are downregulated relative to some normal or to other people, etc. Well, that cell state changes, right? A minute later, there'll be a different set of transcripts that are up or down. If you stress somebody, or you give them a drug, or you put them in an oven, their transcriptomes will change. So what do you mean exactly a transcriptome, right? There's an infinite number of human transcriptomes, in fact. There's uncountably many, right? So all that has to be tracked somehow, right? And the experimental procedures of what kind of cells, in what state, and what environmental or chemical or perturbagens were present, that all has to be recorded in intelligent ways. And when it gets to proteins, it just gets more and more complicated, right? I mean, we can talk about a nascent amino acid chain, okay, the primary structure. We can talk about secondary structure, beta sheets, turns, alpha helices, three-dimensional structure, the fold. But then, you have dynamic processes, like the folding process itself, how chaperones come in and assist the protein for folding, block certain steps of the folding process when it's wise to do so. You have binding between proteins. You have post-translational modifications that come in and change the structure of a protein, whether it's phosphorylated or not. Certain sites open up. So you need to store all of those dynamic events, right, in databases, and make them accessible. Because our goal is to really form a comprehensive pan-biology tool out there. And there's a lot of noise in the protein world, and excuse me, and that has to be considered. It doesn't really stop at proteins, so you can get into pathways and networks, and now we're really talking about how ensembles of proteins affect other ensembles of proteins, how transcription factors upregulate large responses in cellular pathways, etc. I'm talking about feedback and concerted behavior, right? And the sort of holistic systems biology event. And so it really doesn't stop at proteins, right? There's a lot more to model. And I guess, what I'm trying to get at here is the take-home message is that the data is big. There's a lot of it. It's diverse, right? It's diverse. It's many different types. And I've only scratched the surface here, right? It's dynamic. There's a lot of, in biology, we want to capture that dynamicism, right? Proteins aren't rocks that just sit there, fold ones. It's not, it's something that's moving and jiggling and conforming. And there's a lot of uncertainty in our measurements, and those databases have to reflect that. Okay, so, but you can see kind of in the end, right, what is the holy grail of bioinformatics? And I think it's basically to take an organism like human or yeast or whatever it is, and have this information resource that is like molecular anatomy, I guess, right? That you can go in and say, here's how everything works, right? Here's the model of how everything fits together given what we know, what data has been generated, right? Okay. Yeah. So on that note, I mean, that's a very abstract comment. It's a bit abstract, right? It's a guideline for conversation. Okay. So I thought I would. I'm not sure that this is the right spectrum or compass, but it's a start. And we can talk about this in the lecture if you'd like, if you disagree or add to it. But I would say that bioinformatics resources lie along this two-dimensional spectrum. Okay, on the one hand, you have entities, databases, and they range from being repositories to very highly curated resources. So by repository, I mean that there's very little curation. The people who run that database simply accept data in, and they tag it in a way that, with a unique barcode, for example, and they store some, minor, or like not say minor, but they store some basic information about that data set, who generated, where, etc., what it's supposed to represent, and they make it available for the community. And that's very important. So it's a permanent data dump, right? And that's very different. If you think about being in the lab right now and you generate data, you're using government tax money often, so the people paid for it, and it sits on your hard drive when you're finished, and that hard drive eventually ends up in the being recycled. If it's never, the results in that data is never shared with the population, it's a shame, right? It's a waste of resources. And so, what do you do with it then? What's your sunset clause for your data? You could put it on the cloud. It would be a little bit better, I guess, because maybe your supervisor will find it, as compared to your laptop hard drive, but still not optimal, let's say. Or you could put it into a public accessible database, right? A repository. And at least, it's there. And now it's a resource for everybody else in the field to use and take a look at. Now, on the other end of the spectrum, we have annotated or curated databases, right? Where there's been a lot of, usually these databases sort of are built on top of the repositories, right? And they're built by liaising with experts in the field. So here, you're trying to synthesize data from multiple sources often, okay, and put that together into sort of a presentation of what we believe is the state of the art for whatever object we're talking about, like a gene or a protein or an organism, the genome of an organism, okay? And we'll see some examples of that. But, the curated databases are, I want you to understand the fundamental difference between this end of the axis and this one. Repositories, it's like a data dump, whereas a curated database is more of a specialized, high quality, synthesized exercise. And bioinformatics isn't just about databases, it's about the tools that let you access those databases, right? It's the software. And on the one extreme, you have things like data portals, which are simply ways of making data available in its most basic form, which is that you go to this website and you can click on what you want and you download it. It's like shopping, basically, okay? And that data is then transmitted to your machine in one way or another, okay? On the other end of that spectrum, you have tools that allow you to really do data analysis at the site. So that might include running computational biology tools. So there may be, let's say, proteins there that you can predict their structure with, for example, or even data science, so that you can play with those data sets and move things around, etc., and try and drive, your own analyses using their tools. And that's very nice when they exist. There's not too many of those around. And they might allow for things like visualization as you look at your data. So we'll see some examples that make this a little bit more concrete, but I think maybe try to keep that axis in mind. Let's look at some examples. Okay, it's actually the morning of the day after. I'm still wearing the same shirt, but I have moved. So we are about to talk about some bioinformatics resources out there. The first I want to start with is gene cards. I think gene cards is a really excellent resource for human genes. It's only for human, but it's really well put together. And if you're trying to get a quick idea of what a gene is, it comes up in your analysis, this is I think the place to start. As we work our way through the examples, we might consider our little compass from the previous slide. of where different resources sit. And so what I would say here is that gene cards is really well annotated. It sits close to this sort of end of the spectrum down here, and it's mostly a data portal with not really much room for analysis. So I would put gene cards somewhere down here, but I'm happy to be, we can talk about it in the lecture time, and convinced otherwise. Okay, so what is gene cards? Well, let's explore it. This is the website for gene cards, and I'm really interested these days in a gene called Spdef, and if I click on that, I get a list of different options. I think very clearly our Spdef is right here, the first hit. There are some Spdef related things. This is a long non-coding RNA. It might be interesting. It probably overlaps Spdef, or maybe it's even known to regulate it. I'm not sure. But we start in here. So small description, category. I'm not really sure what GIF-TS is, or the GC ID. This is an ID for gene cards, and the score is some kind of measure of how well it matched my query, which is kind of trivial here because it's just Spdef. So when I come into the card, the gene card for Spdef, I can see lots of nice things all put together. There is some research products in here, some advertising, etc. But sort of spelled out here, quick jumps. But let's scroll our way through. An issue for many genes is that there's multiple names for them, and some genes were discovered independently by different people and worked on by different people. Usually the first name is the official name, but if there's any doubt, this HGNC is the Human Genome Nomenclature Committee, and that's basically a body of individuals. I'm not too knowledgeable about who's on that committee, and they decide, they're the definitive source for what the names of genes are. So if I go and click on this, I leave, and I can see now I'm at a different website, gene names, and the approved name here is officially SAM pointed domain containing ETS transcription factor. So we know it's a transcription factor, and then there's a symbol, etc. Okay. Let me close that window here just to keep the confusion down. So that's common, and it's a problem because sometimes there's a lot of different names. You can see back here an entree gene ID that links back to NCBI, and we'll see NCBI a little bit later today. Ensemble, which is like the European version of the NCBI, which is in the United States. OMIM we'll talk about later, maybe not today. And UniProt is another. So these are all different identifiers to different databases. And you can see that this particular gene has been through the ringer, so it's been updated a number of times. That happens, right? As information comes in, we change and change, and so you get multiple accession codes. That's essentially what these are identifiers. So already you can see that this is not a repository, right? They are curating information. Somebody built this, these different summaries. Now this one comes from RefSec. We'll talk about RefSec today. This is an NCBI entry, so it's linking back to basically a repository. But here you can see this is the gene card people have actually made this summary here. GeneWiki is another site that's useful sometimes. It's a bit like gene cards. Overall, I find it's not as well developed as gene cards. I'm not sure what the Monarch initiative is. So those are things to explore. As we kind of scroll down in here, what you can see are the promoters and enhancers for Spdef. So like I was talking about in a couple slides ago, when you want to start to describe those regulatory relationships, the binding sites, here, all sorts of informations about how the expression of these genes are controlled. Gene cards is trying to incorporate this here. So there's already something there about its regulation and its transcription. The genomic location, these are basic information. It's on chromosome 6, at least according to a certain assembly. You see how it's been documented. It always states not just the position of that gene, but relative to which assembly we're referring to. how long it is, basic facts. And then it links to other databases. We're going to talk about the UCSC Golden Path next, so I won't go there right now. But actually note that this all the information in gene cards kind of ends up in the UCSC browser. So these databases are highly federated, highly integrated. RefSec this is now a reference to the NCBI database that's coming up. Okay. So it's a repository where that sequence originates from. So basically here, GeneCards does all the due diligence to show where that sequence is coming from, right? So not just information about the gene itself, but it gives you a way to trace back about where it was found, where it's stored, its history. Okay, and you see that there are now links to external resources to show the structure of SPDEF. I won't click on those now, but they're different databases where the crystallography or the NMR machine has determined the structure of that protein, okay, in 3 dimensions. Another database is, or databases, UniProt and SwissProt. These databases, which we'll see later on, are pan-organisms, so across the tree of life, not just human. But they only focus on protein-level information. The mandate of SwissProt is across all organisms. This is just human. But the other thing is that SwissProt is only focused on protein. It's not focused on transcripts or regulation or any other features of that entity called SPDEF, right? Just the protein level. So GeneCards is sort of taking what it needs from SwissProt, but giving you a lot more. But the reason why all these links are here is so that you can go back to the original source for which they were derived. Gene ontology is a very interesting entity, and we're going to look at that in part 3 of the course. Both what it is and how to use it and the statistics involved in what's called enrichment analysis. Ontology is a funny word. What does it mean? Intuitively, the mandate of Go, it's called, is to label everything in biology with its function, what processes it's involved in, and where it's located in the cell. Every gene, every protein, everything basically has a place in the world of the cell. And Go was really, in a way, an early example of citizen science. Okay, it was more like scientist science or something, because it wasn't your average citizen. But put it this way, if your whole life was spent exploring SPDEF in the lab, then Go would turn to you and say, hey, we could really use your help to label the SPDEF entry in Go with what, the state of the art in terms of its function. In this case here, it's basically known to be a transcription factor, where it's located and what processes it's involved in. And so Go was basically, or the consortium of Go, was basically asking the world of experts to come in and annotate this computational database so that everybody would have access to that kind of knowledge. All sorts of things. Then we're back to science, the localization for SPDEF, so it's claiming it's nuclear with a certain. Now here it's interesting because it assigns confidence scores to how much it believes that it's in certain regions. So there is some evidence, I guess, that it could be perhaps secreted into extracellular space, given it has like a confidence of 2. Perhaps it's a transcription factor. It's hard to see what it might be doing in extracellular space, but who knows? A little bit more about where on the cellular component size, what it's involved in, nuclear chromatin, etc., and the pathways it's involved in. And we start to get into these kinds of networks that we're going to talk about a lot in the course in different contexts, what it's related to, and that definition, we'd have to define what related means, but all sorts of ways to explore how SPDEF fits into the cell and what its, sort of conceptual neighbors are. This is kind of a nice thing to have too, to get a quick idea of what SPDEF does. So you can see here, this is the expression for this gene across different tissues in human. So you see, for example, this is RNA-seq, microarray, which is a bit older, and SAGE, which is ancient. But you can see that it's pretty ubiquitously expressed in almost all tissues, maybe not the thymus, although the microarray suggests maybe it is. The small intestine and the colon, it seems highly expressed. Salivary gland, it is highly expressed in the breast, right? So, and you can see here, like I said earlier, that it seems to have a role in prostate. And that's why maybe it's been studied in prostate cancer. But for my purposes here, it's the interesting thing is possibly that it is expressed in the breast. Okay, and then back to some clinical information, what polymorphisms are known. So a polymorphism, or a CNV, this is a copy number variant actually. These are the polymorphisms here. So there's a variation from G to A in certain kinds of disease. This case here, when I say benign, not provided, I assume that if I was to link on the SNP ID, it would say it's some kind of probably a cancer. Other type structural variations, we're going to look at those more in the course as we go along in the context of cancer. And finally here, what you get then are some of the most, probably not necessarily the most recent papers, but the ones that probably are considered the defining papers for that gene, the ones that first identified it and named it. Okay, and this will give you a PMID that takes you to PubMed, which is at the NCBI, and we're going to get to there today. Okay, so yeah, I think that hopefully I've convinced you that there's an awful lot of information at gene cards. So I definitely think that, definitely we're at this end of the spectrum with gene cards. Now, you might have noticed that there's very little capacity to search or ask questions about the gene. You're sort of just given this information, right? It's static. And so I would put it more towards the data portal side. I'd even maybe move this up a little bit towards a little bit more towards that extreme. Okay, but it's definitely highly annotated. Okay, the second repository I wanted to introduce you to is called the University of California Santa Cruz Genome Browser. It's not just for human. It can be. It does have genomes for a considerable number of species, mostly model organisms. I guess I think it's safe to say, but not all organisms, right? Not all. It's ones that are generally well studied. What I really like about it is that it brings a lot of data together in a pretty graphical way, in a way that's complementary to gene cards. So I use it a lot to get a feel for the genomic information about a gene. Okay, so it doesn't give you much information about protein or protein structure, right? You don't get too much of an idea of who's regulating it, etc. But for basically explaining where it is in the genome, what the gene looks like, and what its promoters look like, etc., it's excellent. So, okay, so here I've actually already typed in Spdef to this location, and it's brought me to a view of my favorite gene right there. And I can see its neighbors upstream and downstream, this Paxin1 and IL-RUN. I don't know either of these genes, but it would be interesting to investigate those at some other time. I can zoom out or zoom in. If I zoom out, I'll get a more global perspective of this region on chromosome 6, the P arm. And if I zoom in the other way, I'm going to get closer and closer to the single nucleotide base level. So here I'm slightly moved in. I can't see my right neighbor anymore. And I can start to see the structure of the Spdef gene. This horizontal bar denotes the region of the gene. Okay, this bar here is a start site, the beginning of the gene, and then you have several exons as you walk along. So these are the coding regions, and then these regions inside are introns, and they don't code for the gene itself. Now you could ask, why is there two copies here of two rows? And that's because that in the database that this represents, these are from the NCBI, there's probably 2 entries, and they probably have slight differences, either because of sequencing error or because maybe actually they're from 2 different humans. Then there's polymorphisms between them. I'm not sure. That would have to be investigated. But it's basically telling you that there's more than one file for SPDEF on record. And the nice thing about the genome browser, I'm going to scroll down all the way first, is that you have a huge amount of information that you can either ask for or not. So what is this? Okay, you know, I can ask for example, all the information about mapping. I could go back to HG19, which is the previous version of the genome, and ask if there's been differences or updates. I can ask for all sorts of things to do with like the parameters of sequencing in the assembly. That's pretty technical and boring. I mean, I'm not really too interested in any of those things, so I just close it off. I can ask for the genes. For example, those 2 entries up top, they're from RefSec, which is in the NCBI database. We're going to talk about that later today. But there's all sorts of other ones I could ask for from different resources out there. And I can't pretend to know what half of these are, even 80% of them are. But the phenotype in literature, so you notice like a lot of the information for this is also at gene cards, but there's some differences, right? And the way it's presented is different. So I actually have asked for the OMIM alleles and the cancer gene expression. These are 2 disease-related databases. So let me see if I can show you those up here. I believe that these are the OMIM alleles. Basically, I think for this guy SPDEF, there's nothing on record, so it's kind of boring for the OMIM. But for Ccga, which is the cancer gene expression, now it shows me here for SPDEF that how it's expressed across different tissues. I can see that's it's active in the uterus, so it's highly expressed. This guy here is the prostate. Okay, so that's consistent with what we talked about before. This is a brain glioma, and I would imagine breast is somewhere in here. It would be a little bit nicer, if it's ovarian. Ovarian's very related to breast. It's both hormonal cancers in women. That's uterine. So I'm not sure where the breast is in here. It would be nice if it was labeled a bit clearer. These are, I guess, other alternative databases that I'm not sure about. It's always a question of, there's a lot of information on these pages, and so it takes a while to kind of dig in and understand everything that's here. We talked, remember in our presentation, we talked about going from DNA to RNA, we have transcripts and isoforms. So how is that present here? You can see that there's different isoforms for this SPDEF. So this guy here is using the full length of SPDEF, but these guys are truncated, so they've skipped this last exon. Okay, so there are actually different variants of the gene, and at least 1, 2, 3, 4, 5, 6, 7, 8, 9, around 10 or 11 recorded variants in the literature. So probably in different cancers or different patients or whatever context, people saw different transcripts popping up. Okay, and for example, there's some nice little graphics in here. This is from the SEIB, the Swiss Institute for Bioinformatics. You can see how the alternative splicing patterns, which exons are skipped or not. And it kind of goes on and on. So you can ask, if we skip down here again, there's all sorts of other information you can ask for. I haven't shown nearly everything here. I can ask for the comparative genomics, so I could ask that I see mouse and other primates, monkey, etc., to see position by position in the gene what's changed and what's been conserved. I can look at SNP databases that have been collected in populations, differences between populations or in disease scenarios. And I can do things that are quite technical, like look for repetitive regions in my genes, lines and signs and retrotransposons. So it's similar in some ways to gene cards. Other ways, it's just kind of like diving deeper into the genomic structure of that gene and where it's located, etc. Whoops, I lost power, but I'm back. Yeah. So it's similar to gene cards, but it's not as comprehensive. It rather focuses on a deep dive into the genomic structure of the gene, where it sits, etc. Okay. So the next example, okay, so actually then we can go back to this and say, let's play this game. So where are we in that spectrum? Is it a database? Is it curated? I would say it's definitely along the curated spectrum. Okay, maybe not quite as curated as gene cards. Just an intuition. Is it closer to being a data portal, or does it the capacity for data analysis? I don't think it offers much data analysis, right? You know, there's not really any way to run things on what you find. It's very just visual. So I would say that it's really kind of closer to a data portal, and I would say maybe it's kind of like not quite as annotated as that. So I might put the UCSC somewhere up here, right, as a tool. All right. So if we looked at the SGD, there is actually a little bit of capacity for data analysis, right? And it's highly curated. So I would put it, you know, closer, a little bit below this line for the first time. That's maybe SGD, right? Certainly, curation is all about the curation, I think. So the next resource I wanted to talk about was the JGI Genome Portal. JGI stands for Joint Genome Institute. So this is a collection of schools, Berkeley, I believe, I think Stanford and the Lawrence Livermore Labs, amongst others. It's really, what's interesting about JGI that's novel about it is that it's not just about one organism, but kingdoms of organisms or taxa of organisms. That's number one. And that's, if you look here, there's the integrated microbial genome, so it's mostly about bacteria, and metagenomes. We're going to talk about metagenomes a lot, so I won't go into the definition for that right now, but think of bacteria and microbiomes. It also has a database for all fungi, for algae, and for plants. Okay, so JGI is not really about humans or mice, etc. It's about, it is limited in its scope. And I can show you this slide here is the microcosm. This is the genome database for fungi. So if I was to, now what's nice is that there's like good level of curation of this information that makes sense. And if I go over here to the website here, and I travel down to Ascomyda, and I travel even further down to I guess this would be a genre of Saccharomycina, and I click on it, I can see that there's actually quite a few different organisms in that category, including my favorite Candida albicans. And if I click on that link, I'm taken to a Candida albicans specific website where there's quite a bit of annotation about that specific species. So why is that important? Well, now we can do analysis. Well, JGI brings all the fungal data together in one spot, so we can do nice analysis across fungi, not just in one specific organism like Saccharomyces cerevisiae. So that's the first thing about JGI that's interesting, is that it supports this, not just one organism, but many. The second really interesting thing about JGI is that it gives this opportunity to upload your data to the site and run that data semi-automatically through computational biology pipelines for gene finding, for abundance estimation, for phylogenetics, for pathway analysis, etc. All sorts of analysis. It's a suite of analysis tools. So we talked about these databases here for everything from microbes to algae. it also has this genome online database system where you can upload your samples. And there's basically, right now as it stands, there's something like 46,000 different studies that have been uploaded to the system. Okay, so it's pretty big. And in a way, it's catching up to the NCBI. That's the perennial database for storing this kind of information very quickly. Now, here you see you can upload your data, and then you can annotate it with the microbial pipelines. We'll look at that a little bit later in the context of specific data sets. But the third thing that's interesting about IJGI is that you have access to everybody else's studies once they make them public in a really nice way. So for example, if I click here on biogeographical metadata, and I use the map because it's kind of fun, well, I'm really interested in the Caribbean because the Caribbean is warm, especially in winter here in Montreal, and I like warm things. So I can come in here, and I really like Tobago too, because it's one of the wildest islands in the Caribbean. And if I come in here, I can see that there's been 2 studies in Tobago. And if I click on this, I can see there's been a study of the coral viral communities here in Tobago, and of bleached corals. And if I click on that link, I get all sorts of information about that particular study. And including, it says that, there's been, here, the number of sequencing projects has been done. So there's sequencing data available for that project. And that takes me to this ID, and I know that it's metagenomics data. And eventually, if I was to keep following this along, I would be able to download that data because it's been made publicly accessible. And I mean, that's really awesome, right? It's well organized, and it's not just, I guess the thing is, it's not just a data dump, right? People load up their studies, but it's asking for all sorts of information about those studies. And here you can see they've been forced to fill in the time the data was added, but also location, the descriptions of them, all sorts of information about the system that they were polling. So that makes it really a nice way to explore. And you see biosample source, environmental data about everything from the temperatures to the depth, etc. Even things like bicarbonate or iron or nitrogen in the water, all those kinds of things. So JGI is a very nice set of tools. So again, 3 things is that it organizes like microbial, fungal, algae data in a nice way. It allows you to second thing is allows you to upload your data and push it down automated pipelines that we'll see a bit later in the course. So you don't need to really do the analysis at some level yourself. You can trust their pipelines. And the third thing then is that you also have access to everybody else's studies from all around the world. And that, of course, is really nice if you're exploring, okay? Okay, so I think maybe we should go back to our little map here. So where does that sit, along this spectrum? Well, I would say it's not really a repository. It combines that kind of basic data dump with actual curation by asking all sorts of information about those samples. So that's quite good, and it does actually, offer a lot of capacity for data analysis. So, I might put, the JGI down here, and that's a pretty nice place to be, I think, right? So it offers some nice tools. Now, you can't do any analysis, right? I think that maybe it's coming clear that you're only going to score perfect for capacity if it's in a tibble in R, right? Because that's where you have maximum capacity to analyze. But JGI, you have a range of things that you can do to your data that's typical in say, genomic or metagenomic studies. The next one was TCGA. Okay. Well, this one's kind of close to my heart because of cancer. TCGA, the Cancer Genome Atlas program, this is US based. I really invite you to read a little bit more. I think you already have done some of the readings earlier in the course related to that breast cancer data set, which I took from TCGA. TCGA, it's focused on cancer. So this is now the first example we've seen of a database that's not focused on an organism or a taxa, but focused instead on disease. And in TCGA's cases, they've looked at something like, I guess, about 33, I believe the number is, different types of cancer. And so all the cancer data is in one spot. And that's really powerful. Just like having all the fungal data in one spot is important. Now we have all the cancer data in one spot, so we can do pan-cancer studies. And that has a lot of potential to it. Pan-cancer papers are very hot right now. The idea is that some drugs that work in some cancers should work elsewhere, and trying to repurpose drugs, for example, is a nice strategy because you save developing new ones. Okay, so we can go to TCGA. I think I have it loaded up here. There it is. Okay. I can come in, and I think that the way to do this is you go straight to the portal. And I want to say a couple words about the portal. That should be here. Okay, so this is the Genomic Data Commons portal. That Commons, it's a word that pops up often. We're sharing. Okay, and the power here is that I can get data relatively easily. It's not trivial to get the data, but at least a portal, the idea of a portal is to allow people from outside to sort of come in and ask for a part of your big data set and not necessarily have to take it all. It would be hugely wasteful if I had to download all cancer data just to get the breast cancer data set. And so portals are designed to make it easy to download data. So that they're very fundamental in bioinformatics, and a well-organized portal is important. So for example here, projects, I might be only interested, I'll show you kind of the example here. Maybe I'm only interested in breast, of which there's 12 projects. And I can scroll down here and say, well, I just want the TCGA data. There's 2 breast cancer TCGA efforts. And then I might come in here and say, well, I'm only interested in the RNA-seq data, okay, and of which there are 2. There's a lot more options there. And when I do that, I get a list over here of 2. One's called TCGA BRCA, and that's the one that we used in the course. You can see that the numbers are around 1,000. And in particular, we took this transcriptional data expression here, 1097. And there's another data set, TCGA DLBC, and I don't actually remember exactly what that data set is, but I didn't use it. And I can then say, okay, I want the TCGA, and it comes in, and I can drill down, and I can specify what I want, and at the end of the day, I can download the clinical data, which you have, and I can download the actual gene expression data pretty easily to my machine. And then once it's on my machine, the question is, how do I then get that data into a tibble in R? And that's the wrangling procedure. Okay, that leads us to the mother lode of all resources called the NCBI. This is the Death Star of bioinformatics. In some sense, it's the place where it's the master, right? It's where you collect any kind of biological information. This is where you would submit it, or to one of its affiliates in Europe or in Japan. Okay, but they're largely in sync these days. So now the NCBI is very much a repository. Okay, so it collects data, but it also has this interesting function of collecting software. So I want you to do a small thought experiment. Imagine you're a computational biologist just graduating with your PhD. You have a new gene finding function, okay, software. But really all you want to do is surf the perfect wave in an endless summer, and you're never coming back. What do you do with that software? You certainly can't sit on your drive, and you probably published a paper or 2 about it. But that's not really the software, because operating systems change, computers modernize, And so somebody has to do some work from time to time to keep that software up and running. Now, at the beginning of this whole bioinformatics experience, I think the NCBI saw its mandate as perhaps maintaining those important pieces of software for the community. And it does do that for some select pieces of software. Probably the most classic computational biology effort is BLAST. Okay, but by far it doesn't keep track of all software. There's other resources these days that do that as best we can. For example, GitHub, which we'll talk about later. But the important thing here is that the NCBI is a repository for all kinds of biological information and tools. And I think that if you've already done the reading in the NCBI's user's handbook, it's very short. Okay, so let's start to delve into that. Okay, so maybe the place to start is with Genbank. Okay, because Genbank, it's mostly about sequence information. It's along that DNA to protein axis that we've been talking about. But there's a lot more in the NCBI besides this. It's a good skill to be able to parse such a file, like the locus. All files begin, Genbank files begin with a locus. We can see that it's a DNA file of length 5,000 base pairs. This locus identifier, it gets really confusing quickly because you see that there's this locus identifier, there's this accession here, there's this accession version here, and there's this thing called the GI here. And it just gets crazy about all these identifiers. It's just kind of insane. So here, let me try to simplify this a little bit. The GI is like a generic barcode that every entry gets when it walks into the NCBI. When you submit something to the NCBI, the first thing it does is it gives us this number, 1293613, and that number is entirely meaningless, okay? It doesn't mean anything. It's just that no other object in the NCBI has that number, okay? And if I modify this file in any way, then I need a new GI. That's as simple as that, okay? So GIs just are simply unique identifiers. A locus, I honestly don't really know what they are anymore. I think that they once had, they were supposed to like encode in that phrase, like SCU, for example. Here, let's go back. You can see that there's SCU 4985 here, right? That's the locus. That's supposed to be saying Saccharomyces, blah, blah, blah. But it's long lost its meaning. I'm not actually sure why they even keep it around anymore. The accession number, that's really important, okay? Now, there's two things here. There's the accession number and the version, okay? And they're exactly what you might think. I think it's kind of obvious, is that the accession here, where is it? The accession number, that is unique, okay? U49845. Now, that code U actually does have some meaning. I'm not sure the 4985 has meaning, but it's been modified at least once, and so it gets a 0.1. So I want you to actually think, why is that important, right? Well, imagine, I submit I sequence something in SAC, and I submit it to the NCBI, and then 2 months later, somebody says, oh no, Mike made a mistake. Here's the updated version. This is what happens. It still has the same accession number, but it's been updated with this 0.1. Every time, though, your GI number just gets attached, and this GI number and the original GI number have no relationship to each other. Here with the .1, you see that clearly. It's an update, okay? So the biggest challenge of these data sets is really these different identifiers. And once you kind of master those identifiers, you start to get it gets a little bit easier. So here, the accession number, it can be indexing into different databases depending on whether it's genomic or DNA, RNA, protein. It depends on the nature of the object, okay? So for example, I believe it's like a RefSec is another database. You can see it's always the same start of those codes. It's 2 letters by an underscore. So if it's NT underscored, it means that it's genomic DNA. And if it's NP underscore, anything with the P, we know it's an amino acid sequence. So those codes kind of give you a hint as to what's the actual data is. Okay, so accession numbers are really the meat and potatoes of NCBI in terms of indexing. I guess that would be a take-home message there. We go back here. What else is in this folder? What else is in here? Again, think a little bit about gene cards and the UCSC and compare. What's kind of so it has some information here about what organism it's from, the phylogeny of that organism, right? That SAC is a fungi. The reference of the people who made that first discovery of this gene or genomic region. Terry Romer, for example, is the person who did that. And then basically there's information about the entry itself. So for example here we have the source, which is SAC. The chromosome, it looks like it's on the 9th chromosome. Map is probably some assembly of that genome. And then it says where it starts, okay, and this is the translation. And you can see here now that there's a reference, an accession number to a protein, okay? And so this amino acid chain is part of a larger protein, and it's giving the access to that. And you can see that protein has both an accession number and some GI that it was given when it first entered. And that's the first part of that gene from the first to 5028th base. This is a bit more about that gene in terms of from 687 to 3158, and it comes in here and says a little bit more about what that's specifically doing. Okay, so basically at the end of these files, it's breaking down the sequence into its constituent parts and explaining those guys individually. Okay, so that's that file. Now it changes depending on whether you're talking about genomic DNA, genes, proteins, etc. But let me put it this way. If we go back to our image here, basically everything that comes into NCBI in the Genbank gets such a file, okay, and it gets such an accession number, whether it's a genome, plasmid, gene, transcript, or amino acid chain, okay? I hope that's clear. So it's kind of the basic unit in a sense of now it's not like gene cards. There's no interpretation of it, or very little interpretation. There is gene names, etc., but there's really no analysis or visualization associated with it. It's really the raw data. Okay, so an important part of the NCBI is the search engine called Entree. It's like Google for the NCBI. And I can search on SPDEF, but let me come over here, and that's, if I go to the NCBI, and if I type in SPDEF here, I will get back a very informative page, because it searches across everything, all of its databases in the NCBI, and this is kind of a good way to get to know the NCBI actually. So, for example, SPDEF showed up in 7 books. Okay, so NCBI also houses medical literature, life science literature. PubMed are all journal papers, and PubMed Central is includes, for example, conference proceedings and abstracts. It's a bit more general than PubMed, and these are incredibly important tools, right? PubMed is, if you haven't used PubMed by now, you will if you stay in science. It's SPDEF shows up and 351 genes, so we can take a look at that. That seems kind of ridiculous, right? How could it show up? And that's because probably some are mouse, human, mouse, rat, zebrafish, cattle. So basically now if I click on any of these links, I get the SPDEF in that organism, right? And all sorts of information about it. At the protein level, I can see that there's like 590 different proteins reported for that across those genes. There's one structure known for SPDEF. If I click on that, I can see that's what it looks like in 3-dimensional space. Okay, so that's a nice resource too. Oops, sorry about that. I can get what the genomes where it shows up. For example, It's been recorded in 82 genomes. I guess the first one here is possum, is that right? Yeah, opossum, mouse, et cetera, et cetera, et cetera. So it takes, you can quickly find out, you can zoom in on what genomes it's in across the tree of life. The genetics, so this comes back to polymorphisms and mutations in disease. And I could click on, say, clinical variables or ClinVar, and I'll get basically all the times that mutations have been seen in SPDEF. In this case here, this is a G to C transversion at site 504. Transition, and you could probably dig down into what that mutation is actually about. And there's a lot of other information available for SPDEF that is fun to explore. Okay, so then where are we then for the NCBI? Well, it's definitely a repository, and there is some capacity for data analysis, so I might put it down somewhere in here, and that's the NCBI. Okay. All right. So to conclude here, I gave a long list of points of reflection that delve a little bit deeper into some of these issues. I invite you to take a look. I think that they're kind of just thought exercises for the most part. Of course, there's literally thousands of resources out there for bioinformatics, and there's no way to cover them all. The NCBI itself is really a lifetime of exploration unto its own. It's a good starting place, though, and I recommend that we keep exploring all these tools as we move through the course. Okay, thank you very much.